everybody know, my name is Nigel. Like Jeremy said, I attend the University of Wisconsin Stevens Point. I double major in biology with an emphasis in genetics and also wildlife ecology research and management. This year I'm a junior, so for me not that much time has passed because I'm still in my undergraduate. I want to start off with my academic history and my involvement. This year I am serving as president for Black Student Union. We put on a bunch of educational programs for our community in central Wisconsin. We just actually put on a soul food dinner, and we had 233 attendants, so it was a really nice event. I'm treasurer for Manners, that's Minorities in Agriculture, Natural Resources, and Related Sciences. We strive to promote diversity within the natural resources. And then I'm also a student member of the UWSP CNR Undergraduate Symposium Committee. And I've also been working on the Inclusivity Committee, trying to promote inclusivity on campus. That's basically tolerating everybody's differences, regardless of ethnicity, socioeconomic class, and other things. I'm going to get into my summer experience. Just this past summer, I was in the Partnership Education Program with Dr. Gerald and Angie. This is located in Woods Hole, Massachusetts. I had the opportunity to work with NOAA, HUI, and the Mashpee Police Department. My research project included working with didenum. I was looking to identify a native species that would prey on it. It is an invasive species. There are economic and ecological implications for the didenum. The local Wampanoag tribe out in Mashpee has an oyster farm, and because of the didenum, which attaches to firm substrates, it can overcover the oysters and prevent them from filtering their food. So this is what it looks like. It's kind of really slippery when you get a hold of it. But basically what I did to try to identify its predators, I attached it to these plastic plates, and I set them in a water tub, which had continuous water coming in from the ocean that was right there. I used 14 local organisms. I used green crab, spider crab, blue crab. I also used several different species of fish. I used trout, and I also used toadfish. And so the way that we identify whether or not these predators actually ate the didenum is we set them inside the tubes with the didenum as well. And so I would photo document every day whether or not there was some progress as whether or not it was being eaten. And so what we wanted to look for were the spicules that is made up of the didenum. Didenum is made up of sodium carbonate. And so when you look at it under a microscope with a polarized filter, you get this fluorescent sodium carbonates they look like little mines in the water. And so what we found was these huge blocks of sodium carbonate instead. The one organism that we saw markedly decrease in the amount and size of the didenum was the spider crab, and what it actually turned out to be was that it had ate its own shell that it had shed. So a little bit of that was inconclusive, but I know that they're trying to do that again. This is my current internship. I'm looking at the seroprevalence of free-ranging white-tailed deer in Wisconsin. That's out of 2 sites, Winter and Shiocton. The differences between those two is land type and also the density of deer per square mile. We achieved this by going out and trapping them in box and clover traps, and then we obtained blood samples through the jugulars. So that's a 3-year project. I have 2 years of data right now. Right now I'm currently going out and still deer trapping and obtaining blood samples for them to be sent off, and so I can do testing on it. As I said, I actually recently added the emphasis in genetics this year after the summer spinner program with PEP. Right now I'm looking at yellow lethal on the soybean genome. Yellow lethal is a mutant gene. It causes the plant to turn completely yellow, preventing it from photosynthesizing, effectively killing the soybean. And then I also recently just started this project, and we got funding from Iowa State University for it. It's called SGR, the state green protein. So what it does is it kind of prevents leaf senescence, and what that is biological aging. So what we usually know for plants is with chlorophyll, when the season changes, you usually see that die off of the leaves and them turning yellow. And so instead of turning yellow, they stay green. But what we actually see, we don't see any kind of photosynthesis competence increasing or retaining. It still decreases, but the leaf remains green. And then I was going to go into the methodology of it. So we compare it by looking at a normal gene and then the mutant gene, and we run these on primers, and we duplicate our DNA within the PCR machine. We run these through an electrophoresis gel, and then we image the gel to discern differences between our bands of DNA. And so our gel has phenamine bromide in it, which allows our DNA to fluoresce. And so what we look for is recombination of these genes. We look for polymorphism. So like how you can see here, these are differences in these bands. You have a double band right here, and then you have these two distinct bands where one is higher than the other. So these will be two different things. And that is all I have. Yes.
So what's next? What are you doing this summer?
I will be studying abroad in Germany, Poland, and Iceland. I'm looking at how they manage their natural resources and how we can relate them to how we manage our natural resources over here.
And how long will that be?
July 9th through August 14th. And so in June, I'll be working in my genetics lab on my current 2 projects.
So when you notice that the leaves were chlorotic, or, you know, for those who, like, I guess don't really know the term, basically they lack chlorophyll.
Mm-hmm.
So